what I'm going to be talking about today is some work that we've been trying to do to look at how we might be able to distinguish the identity of different carbon fixation pathways in deep time. So, I mean, we care about this, right, because not only are we taking carbon dioxide and converting it to biomass, but it has to do with energetics, material transfer, and perhaps more importantly, the Lindemann-style trophic dynamics of the global biosphere at the time. And one of the things that you can do to identify which autotrophic pathway may actually be fixing carbon is by looking at the ratios of carbon-13 to carbon-12. It turns out that due to the tension between equilibrium isotope effects and enzymatically controlled kinetic isotope effects, you will end up with, at the end of this process, biomass that's enriched in the light isotope of carbon and leaving behind carbon dioxide that is enriched in the heavy isotope of carbon. Okay? So how do we actually get at this effect, and how do we use it to identify different carbon fixation pathways? Well, one of the things that gave me great pleasure to hear is that I'm going to be talking before Jody, who has one of what I think the most crystal clear presentations on how you actually can go out into the environment and figure out from the carbon isotopic composition of dissolved CO2 or its counterpart dissolved bicarbonate, and then the carbon isotopic composition of primary biomass, in here, particulate organic carbon, and figure out from the isotopic difference there the isotope fractionation associated with primary production. And if you go and read this paper, one of the amazing things about this data set is that if you correct for nitrate and you correct for temperature, you're left with a temporal signal that may have something to do with enhanced CO2 access as time has increased. Okay? So one of the things, though, that we have to worry about is that rocks are not biomass in dissolved CO2. So how do we go from the dissolved CO2 carbon isotopic composition, the composition of biomass, and figure out something from the geologic record? Well, the way that we do it is we approach it in this sort of haze diagram sense, where we've got the carbon isotopic composition of these different compounds on the left axis. Here is what we want, this difference between dissolved CO2 and biomass, but we have to be concerned with the temperature-dependent isotopic fractionation associated with carbonate speciation. We have to be concerned with the mineralogic associated fractionation associated with the different precipitation of carbonate minerals. And then, of course, there's going to be some small but important fractionation associated with the reworking of primary biomass into sedimentary total organic carbon. Okay? So one of the things is that what does this record of mineral carbon isotopic compositions and total organic carbon isotopic compositions look like in deep time? One of the things that we're very fortunate for is that there was recently an incredible statistical reanalysis of the carbon isotope record in deep time. And the carbon isotope record in deep time, it turns out, has two distinct types of states. There's going to be the intervals that we're going to ignore here, so-called transitional intervals associated with climatic upheavals and non-steady state behavior in the carbon cycle. But we're going to be focusing instead on these non-transitional intervals, largely the Phanerozoic, middle Proterozoic, and back in time. So what we want to do is we want to invert this haze diagram then and go from the carbon isotopic composition of sedimentary carbonate minerals, the carbon isotopic composition of sedimentary organic carbon, and go through a procedure that allows us to extract, to the best of our ability, the isotopic difference between dissolved CO2 and primary biomass in deep time. So it turns out that this technique was developed by Sarah Hurley, a postdoc I'm fortunate enough to be working with. And what happens is we go like this. Let's look at a histogram for a certain time period of the isotopic composition of carbonate carbon. And what Sarah realized is that if you do an agnostic resampling of the isotopic effects associated with carbonate mineral precipitation, the temperature-derived isotopic effects associated with carbonate speciation, what you can end up with is you can end up with a frequency distribution of the delta 13C carbon isotopic composition of dissolved CO2 for the time period that you're interested in. Similarly, if you take the isotopic distribution associated with sedimentary organic carbon and run it through a random resampling looking at a distribution that might have something to do with how primary biomass is fractionated during microbial reworking, you can end up with a frequency distribution of the carbon isotopic composition of biomass in deep time. Combining these two, effectively just taking their difference, gives you a frequency distribution of the so-called epsilon p for primary productivity, epsilon for a difference in delta values, that then may tell you something about what carbon fixation pathways were operating in deep time. Okay, so when Sarah did this with the non-transitional intervals in the carbon isotope record, what she returned was the following type of distributions. If we start over here, something centered at about 50 million years ago, going back to something about 250 million years ago, what you can see is a slight increase in the most frequent value of carbon isotope fractionation by the dominant primary producers at that time. Though there are a number of evolutionary events that may be associated with this, it's also possible that what you're looking at is simply the effect of the gross long-term increase in CO2 as we go back through the Phanerozoic. And as CO2 increases, what you end up with is you end up with less of a more efficient operation of the carbon CO2 concentrating mechanisms that are associated with these organisms, leading to a larger fractionation. We're going to focus next on two intervals for a case study. The case study one is this mid-Proterozoic interval. And what we want to do is we want to ask the following question. Was the middle Proterozoic actually the age of Cyanobacteria? What I'm showing here is a diagram that has a bunch of different compilations of oxygen levels from today back to the early Archean. And shown here is, of course, the great oxygenation interval, but also shown down here in green are some indications both from geochemistry and also from microfossils of cyanobacterial oxygenic photosynthesis. And then on this side of the diagram, some information from molecular clocks, from microfossil evidence, and from biomarker evidence for a primary production, global primary, biospheric primary production dominated by algae. And what we're asking is, in this mid-Proterozoic interval, was it really an age of cyanobacteria? Can we use this technique to say, okay, the primary producer at this time, which it seems like it was a cyanobacterial population, or was it something else? So, one of the things that is a complicating factor in this is that all extant cyanobacteria have a CO2 concentrating mechanism. Probably the most famous bacterial organelle is known as the Carboxy Zone. It's where cyanos today have their rubisco, have all their carbonic anhydrase. They bring in bicarb. It gets converted through the activity of carbonic anhydrase to carbon dioxide right at the site of carbon fixation. And what you can do is you can overcome some of the catalytic inefficiency of rubisco in that way. And there have been a whole bunch of different estimates for when the cyanobacterial CO2 concentrating mechanism may have come into play. And what it is usually associated with is drops in carbon dioxide levels. It makes sense. If carbon dioxide levels fall, you would expect that a CO2 concentrating mechanism would be developed. So we decided to test this, and Sarah in the lab was able to create a carboxyzone-less mutant of the wild-type Cyanobacterium Synechococcus 7002. And these green fluorescent images show the distribution of rubisco in these mutant cyanobacteria. She grew them under different CO2 levels, and it turns out that they're high carbon requiring mutant, which means that growing them at very low CO2 levels is very difficult. Fortunately, though, it's a relatively simple system to model, where you just have CO2 diffusing into the site of carbon fixation. So we were able to model what might happen at lower CO2 levels. Even at relatively low CO2 levels, what you can see is that the range of carbon isotopes that are produced by these potentially physiologically ancestral cyanobacteria does not cover more than about 30% of the epsilon primary productivity range that we see in the middle Proterozoic. So what happens when you just take a wild type cyanobacterium and grow it, again, under different CO2 levels? Well, what happens is you produce a range of carbon isotope values that covers 95% of the middle Proterozoic epsilon primary productivity distribution, suggesting then that one of the simplest interpretations of the middle Proterozoic primary productivity record is that cyanobacteria with a carbon CO2 concentrating mechanism were the dominant primary producer at this time. Great. So what I'm going to do now is I'm going to speed into the second case study, which is looking in very deep time and saying, okay, what might this carbon isotope fractionation distribution mean? There's some similarity here between what you see in the middle Proterozoic and what you see in the early Archean. And in fact, that may be used to say, okay, the same carbon fixation pathways were in operation at this time. What we decided to do was a thought experiment, and working with some colleagues at BU, Daniel Segre and Josh Goldford, we decided to look at what might potentially happen if you created a endergonic and biomass-capable metabolism from a simple set of potential prebiotic seed compounds, shown down here: acetate, carbon dioxide, formate, hydrogen sulfide, and some others. And so essentially what Daniel and Josh did is they went ahead and they led to a network expansion associated with this metabolism. And they were able to show that not only was it endergonic, it was capable of producing biomass. And what we wanted to know is, what's the carbon isotopic consequences of this expansion? So it turns out that carbon isotope fractionation is both incredibly confusing, but at some level, relatively straightforward. So it depends on two things primarily. One, the nearest neighbors that the carbon atom is bonded to, and then the nearest neighbors to those nearest neighbors. And so what a colleague, Chris Butch, was able to do was take this full set, hundreds of different compounds, and actually calculate what the isotopic fractionation would be as this expansion happened. Okay? And here, shown on the left, is the iterations. Here are two images of that epsilon primary productivity. On the top, the number of compounds in the network is shown linearly. On the bottom, we've got it plotted versus the log number of networks, a log number of compounds in the network. This one is easier to see the differences. The early networks are shown here in blue and in red. But as the network increases in diversity and potentially in complexity, isotope fractionation actually goes the other way. It is narrowing up and approaching a single mode at about 18 per mil for the difference between dissolved CO2 and biomass. So what's the reason behind this? It turns out that when you count the bonds and the bonding partners that are involved in this, as Chris has done here, what you see is that as the network expands, your overall bonding environment is getting more reduced. You're seeing a lot more carbon-carbon bonds and carbon-hydrogen bonds. And here what I've shown is the distribution of Chris's chemoinformatic prediction along with the early Archean distribution. And though they're not exactly the same, there may be some kinetic effects here and less equilibrium effects here. One thing that's very clear is that an epsilon primary productivity of around 18 per mil or 20 per mil is something that would have been in the past associated with, say, the carbon fixation by the Calvin cycle, may simply reflect the development in these networks of aliphatic carbon-carbon bonding. Okay, I'll leave you now with what we know, what we think we know, and what we want to know, and thanks for coming.